Hello everyone, welcome to Ibdp Biology. Today our topic is going to be membrane structure. So let's get started. Before starting the membrane structure, I would like to explain you with some terms which we use in biology, and you're gonna use it more from now on. Those are the hydrophobic and hydrophilic molecules. What does it mean, hydrophobic? Having an aversion to water. In other way, it means that those kind of molecules don't like water. At first they look like they're dissolved in water, but after some time, after a few seconds, you might see that they create rain droplets in water, such as oil. So when you stir oil with water, at first you may think that it's already dissolved, but oil never dissolves in water, so it creates rain droplets and actually becomes suspension in water. So this kind of molecules we call hydrophobic. When it comes to hydrophilic molecules, they have an affinity for water. They love water and they are easily dissolved in water. And as you see in the picture, the hydrophobic coating and the hydrophilic coating, how they actually dissolved in water. It shows their characteristics. So, our main focus today is going to be the cell membrane. As you know, the plasma membrane surrounds the all cells and it separates the external environment from the internal constituent of the cells. But as you see here, I would like to show you this video how it's actually look like. This is the plasma membrane and how it looks like. The cell membrane is actually the model of the cell membrane is very hard and is very difficult and very unique. So we're gonna just get into detail of how actually the structure of the cell membrane. Phospholipids are unusual because part of a phospholipid molecule is hydrophilic and part of it is hydrophobic. What does it mean? Plasma membrane is made up of phospholipid bilayer. Bilayer, it means it's made up of two layers. It's made up of these circles are phosphate heads and they're hydrophilic. But those tails are hydrophobic and we call it phosphate heads and hydrophobic hydrocarbon tails. So it is actually structured like that. The phosphate heads are facing outside while the hydrophobic tails actually facing the inward. So the phosphates are actually facing the water environment while the hydrophobic tails are not getting into not getting contact with the water or external environment. So, integral proteins. As you see, integral proteins are embedded in the plasma membrane, and these proteins control the entry and exit of the specific molecules from the cell. They also have a hydrophobic and hydrophilic region which helps them keep in place in the membrane. When it comes to peripheral proteins are actually attached to the plasma membrane or attached to the integral proteins. So the peripheral proteins are known as glycoproteins because they have a carbohydrate attached and they function in immune response and are involved in cell-to-cell recognition. Phospholipids, a molecule that is a constituent of the inner bilayer of biological membranes, having a polar hydrophilic head and a non-polar hydrophobic tail, as you see here, the phosphate head and two fatty acid, unsaturated fatty acid, which we call the hydrophobic tails. So, the models of the membranes were developed long before membranes were first seen with the electron microscope. In 1935, Dawson and Danieli proposed a sandwich model in which phospholipid bilayer was lined on both inner and the outside by a globular protein. So they made a model, like we call it the sandwich model, and they proposed that the plasma membrane in both sides, in the external, in the inner and outer surface, they are covered with the globular protein. This model consists of a phospholipid interior surrounded by protein coats on each side. But in 1972, the Singer and Nicholson proposed fluid mosaic model. So, and as you see in the freeze fracture image, the image was a turning point for the model of the plasma membrane. It shows the bumps and grooves on the inside of the membranes, and these bumps and grooves are actually the membrane proteins. It supports the idea of protein embedded within the membrane as opposed to the outer protein layer proposed by Davis and Danieli model. Now, Singer and Nicholson proposed that the hydrophilic regions of proteins and phospholipids are in a maximum contact with water, while the hydrophobic regions are in a non-aqueous environment. So they suggested that it's actually the fluid mosaic model, and the proteins are actually embedded or attached to the plasma membrane. This is how the fluid mosaic model looks like. And as you see, the plasma membrane is not covered with a protein, but proteins are embedded or attached to the plasma membrane in different sides. Why fluid mosaic? A membrane is mosaic. Why? Because proteins and other molecules are embedded in a framework of phospholipids. A membrane is fluid because most proteins and phospholipid molecules can move laterally. So you know that hydrophilic phosphate heads are actually making the plasma membrane fluid, but the hydrophobic tails make it much more solid. So you must be able to draw the fluid mosaic model, which is the current model of the plasma membrane. Ensure you draw each part to scale and include all relevant labels. And your drawing should include a clear drawing of individual phospholipids, molecules using circle for the head and parallel lines for the tails. You must also include a range of membrane proteins, including glycoproteins. So now this is how we're gonna just create a membrane transport model and the plasma membrane. As you see, it's made of the phosphate heads, the hydrocarbon tails, the protein, integral protein and peripheral proteins, as well as the cholesterol is shown here. That you're going to make this kind of project about membrane transport and plasma membrane project. So, as I said, the head of the phospholipid is hydrophilic. The fatty acid tails are hydrophobic. Phospholipids are considered amphipathic molecules due to the fact that they have both a hydrophilic and hydrophobic region. The phospholipid molecules are arranged in the plasma membrane due to the way they react with water. Because the polar heads of the molecules are hydrophilic, they are easily contact with the external environment, with the water, and they're arranged so they're always facing the internal and external fluid environment of the cell. Due to the fact that the fatty acid tails are hydrophobic, they face inwards towards each other and evade from the fluid environment. And remember that hydrocarbon tails, they are attached to each other just because of their hydrophobic, but the phosphate heads are hydrophilic and they're attached to they are much more attracted to water. The fatty acid tails in the center of the membrane are attracted to each other, and phosphate heads are attracted to the surrounding water, as I said. There are many different types of membrane proteins, and they occupy different positions in the plasma membrane depending on their structure and functions. So let's get to know them. Hormone binding sites. So a site exposed on the outside of the membrane allows one specific hormone to bind based on shape. A signal is then transmitted to the inside of the cell. Immobilized enzymes. Enzymes located in the membrane either catalyze reaction inside or outside the cells, so depending on whether the active site is on the inner or outer surface. Cell adhesion. Proteins from adjacent cells may hook together to provide either permanent or temporary connections. This form junctions referred to as gap junctions or tight junctions. This is a cell-cell communication. The cell-to-cell communication is a protein involved in the cell-to-cell communication have an attached molecule of carbohydrate which provides an identification label for the cell. Channels for passive transport. Channels are passages through the center of membrane proteins. Each channel allows one specific substance to pass through from high concentration to low concentration. Pumps for reactive transport. Pumps release energy from ATP and use it to move specific substances across the plasma membrane, and the energy is used to change the shape of the protein. This is the glycoproteins, and it's a membrane protein that is covalently attached to a carbohydrate. They're used for the cell-to-cell recognition and play important role in immunity. So, cholesterol. So, now I'm going to talk about the cholesterol. So, animal cell membranes contain cholesterol, which plays a role in stabilizing and maintaining integrity of the plasma membrane. How? Cholesterol is a type of lipid, but it's not a fat or oil. It belongs to a group of substances called steroids. And most cholesterol molecule is hydrophobic. So it is attached to hydrocarbon tail and in the center of the membrane, but one end of the cholesterol is hydrophilic, and this is attracted to the phosphate heads. So the hydrophilic part is attached to the phosphate heads, hydrophobic part is attracted to the hydrocarbon tails. So, as I said, the role of the cholesterol is very important because the fluidity of animal cell membranes need to be carefully controlled. And if they were too fluid, they would be less able to control what substances pass through. But if they were not fluid enough, the movement of the cell and substances within it would be restricted. So the cholesterol actually regulates the fluidity of the animal cell, so it shouldn't be like neither solid nor like very fluid. So this is the help of the cholesterol for the cell membranes. Okay, the cholesterol will affect the fluidity of the membrane, and it has different effects at different temperature. The location of the cholesterol molecules prevents the close packaging of the phospholipids, and this helps prevent the membrane from solidifying it if the temperature drops. So this is the review part. So you're going to define the plasma membrane structure, hydrophobic and hydrophilic terms, function of the proteins, discuss the function of the membrane proteins, and what's the function of the cholesterol. If you cannot answer one of these questions, go back to the lesson, the presentation, and again reread the all slides, so it's gonna help you. Thanks for watching, guys. Have a nice day. Bye bye.